Um, all right, so I will begin again. Open models, open, weight, models mean a lot to US, not just US, but people in developing, countries and other parts of the world, because it costs, a lot to be able to um, use some of these, models, right? So we all have subscriptions to Cloud Code, which is great, but it's not cheap, right? So how best can we get access to intelligence, for less the cost, right? It costs electricity also, right? So it costs in terms of energy, but there is a new model by thinking machines, which is open in the sense that you could customize it and adapt it to your enterprise or for your specific needs. And I think especially for um, developing countries, really investing into open models will be very huge. I think 2 weeks ago there was OpenAI GPT 5.6, so Terra Luna. I've tried it. I think it's great. The economics are very great in terms of how much you pay versus the outputs that you get. I think the economics are great, and I've personally been using it for interactive, scientific visualizations of workflows and like doing my developments, right. Well, so I think it's something you may want to look into in addition to the cloud science and the Fable assets, which is Fable 5, which is ending this weekend. There was also a news, yesterday. There was this new model, open weight, model by a Chinese company called Measured AI. I mean, it claims to be really great and very like in terms of level of competence to be similar to Opus level, which is by cloud. So I think it's something you may also want to look at, especially if you think about low cost models for your work. Grok 4.5 is fast. It is very 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 fast and cost effective. So if you have access to it, you may want to try. It was this thing I saw, I think, yesterday. I think it's called Schema. So Naval, it's one of the popular voices um, on the internet and YouTube. So there's a harness, by an impossible machines or company he was building, in secret, and he claims to, um, achieve high, amounts of how do you call it? I don't really know how this works. I have to do like really deep investigations into it, but I think it's something to like really put on your radar in terms of open source models and cost-effective intelligence for certain environments, right? So with that, I would want to welcome Ali. Ali is at the Chino Center in New York City, um, and he's gonna take us through embedding, how we can use, embedding. I think, later I would do sort of like a recap of what we've been, studying and where they all, fit in and how you can move, from there, right? So embedding is at the training, level of, the model. And then, after you've, trained the model and you've got your, model, for example, Opus, 4.8, or like, you train your model and you've got maybe something like ChatGPT, 5.6. Um, there is something called, post training, right? These are things, that you do on top of the training, right? So it's being applied at pre-training, post-training. They both have, applications and, usefulness. And Ali is gonna, take us, through different type of model we've been studying or we've been using language models like Opus by Claude, GPT, and grok and the others, right? But then this is a different type of model, and I think it's a single cell model, right? Foundation model. So, without much ado, let's welcome Ali to lead today's session. And I want to say thank you very much for leading the session today.
Thanks, Sam, for the introduction. I'm gonna share my screen. I guess that's up to, um, guessing everyone can see what I have up, right?
I think Paz is here also.
Yeah.
So if you have any quick announcement before I leave, or maybe afterwards, that'd be great.
Okay. Nice. Welcome, guys. Feel free to interrupt and ask me questions about anything. But the title of my talk today is bulk RNA-seq deconvolution, leverage a single cell foundation model embeddings. And I know it's a mouthful, but we'll slowly go through the background of what this is and then how we're using single cell foundation models for our task. So I guess my first question is to gauge if everyone is familiar with the types of next gen sequencing we do in biology or in bioinformatics. So there are two different types in terms of RNA expression data. We have bulk and then we have single cell. So bulk RNA sequencing is basically, let's imagine you have a tissue sample. So maybe someone has cancer and you go in and you do a biopsy. So you scrape parts of the tumor and the surrounding environment, and then you crush it. You mix it together, and then you get RNA, and then you sequence that RNA. You've done bulk RNA-seq, and the reason why it's called bulk is you've combined different cells in that same tissue. You're not separating cells, so that combination of all the different cells in that same tissue is why we call it bulk. And the expression you're getting from there is an average of all the different cells that exist in that tissue that you've collected. Unlike bulk RNA sequencing, there is a newer version of this that has been, I think we've been doing this for the past maybe 5, 6 years now, called single cell, maybe even longer, called single cell RNA sequencing, which is just like bulk, but now you're working at the single cell resolution. So what that means is, as opposed to getting all the tissue together and then mixing them, you're able to break that tissue down into very individual single cells. And within each single cell, you can then, sequence the RNA expression in that single cell. So the
So,
best way,
so if I may interrupt a little bit, could you take an example like of a tissue, and probably we could maybe on top of our head annotate the cell types or like the different types of cells that are in this tissue. Um, and I ask this question, because a lot of us here having really done single cell and even bulk RNA before. Um, if you want to go, that's fine. I could also go, but I was thinking you could pick one example, maybe blood or
no, that,
something, and then
that's a good question. I was
so
gonna
that we can relate it to
what's
like
going on. Yeah,
real, yeah,
I,
right.
yeah, I was going to start with this. I think this is the best way to picture it. So the very popular examples bioinformaticians use to explain this concept is through the idea of of a smoothie. Everyone has had smoothies before, so smoothies are it's a very good example to do it. So a smoothie is a combination of different fruits. So in this picture right here, or cartoon, there's strawberries, there are blueberries, there's some sort of citrus. Here, I think there are apples at the bottom. A smoothie is just you put all this in a blender, maybe add some milk, and then you blend it, and now you have this cup of a mixture of different fruits. if you taste this fruit, rather if you taste the smoothie, um, you can, if you've had the individual fruits before, you can tell that there is, a strawberry in this smoothie. You can tell there is a blueberry in this smoothie. You can tell there is an apple. But what you can tell is the exact proportions of the different fruits you use to make that smoothie. But you can tell definitely by tasting that there's a banana in the smoothie, for example. So this is a perfect illustration of what bulk RNA sequencing is. So the taste of that smoothie, in the case of bulk RNA sequencing, the expression is a combination of the different cell types that exists within that tissue. So this would be our tissue sample, then this would be our smoothie, right? In the case of single cell, as opposed to like having a combination, you separate the different fruits into like its different components. So you have blueberries here, strawberries, you have the I think it's an orange. Let's call this an orange. An orange here, and then you have an apple. So this is the main difference here. So with single cell, you have the individual fruits, and then with bulk, you have it combined. So now let's use an actual tissue example. So let's say someone has cancer, and then you go into, let's say, endometrial cancer. So you go in there to do a biopsy and you scrape out cells around the tumor. When you scrape out cells around the tumor, you aren't just scraping out cancerous cells, you're scraping out healthy cells. And then you're also scraping out immune cells, because usually around tumors, there are like immune cells trying to attack the tumor. So you're scraping out immune cells. If you do a bulk RNA sequencing of that tissue sample you just scraped out, what you're getting is an average expression of all the different types of immune cells you've scraped out, so B cells, T cells. You're also getting the different types of cells, healthy cells, and cancerous cells that exist within that tissue, so within the endometrial tissue. It's a combination of everything, right? So yes, you're getting some cancer cells there. Yes, you're getting some healthy cells there, but also you're getting like the tissue microenvironment. So the B cells, the stroma around that tissue. In the case of single cell, we're able to like separate the tissue from the biopsy into individual cell types. So if we can separate the cells, we can sequence the RNA in those cells, and then later down the line, we can classify or annotate those cells. So we can say, hey, this is a B cell. Hey, this is a T cell. This is an endometrial healthy tissue cell, and this is probably like, a cancerous version of that cell. So single cell does give us more resolution and gives us like more information compared to bulk. But bulk was the first one that was like, done, and it's very cheap to do compared to single cell. That's very expensive. So we have lots of data that's already in bulk across like healthy disease states and across different types of like human diseases. We do have over the past years now, we do have single cell data across different diseases as well. But making more of this is expensive. So sometimes what researchers can do is we can go back to the old bulk that we have and then use some of the newer single cell information that we have and try to combine these two to tell us the proportions of the different cell types that exists within the bulk. The way you can think of this is if you go back to the smoothie example, right? Um, if you taste this, if you've had a banana before, or strawberry before, or blueberry before, you can tell by just drinking the smoothie if the smoothie has more blueberries, or more strawberries, or more apples. This process in bioinformatics is called deconvolution. So we're trying to go back to our bulk, right? Using some signature from single cell to help us break down this bulk into cell type proportions that made up the bulk, purely using the expression data. And in the case of this smoothie, it will be purely using taste. We're trying to use the taste of this smoothie, right, with the taste of the individual fruits that we know to break down the proportions of the different fruits that make up the smoothie. Does that make sense? Okay. I'm guessing that's a yes, but we'll keep seeing more of this. So if you have more questions, you can ask me down the line. But the take home from this is there is bulk, right? And then there is single cell. Bulk is more like average across different cell types, and single cell is individual cells. Um, but the same like gene expression information. And then the process of deconvolution is trying to break down our bulk into cell type proportions using some single cell signature. So we're comparing the single cell with the bulk expression and trying to use that information to break down this bulk into the different cell types that make up the different proportions of cell types that make up the bulk. Sometime around, I think it was 2003, maybe 2022. I think it's 2021, 2022. I can't fully remember. But this was quite trendy. The tumor microenvironment was quite trendy. What that means is, actually, I'll start from the story itself. There was a researcher called Bagev, I think that's how you pronounce his name, that showed that across public bulk TCGA data. So TCGA data is a public data sets of like different cancers that you can have access to. He showed that across the different TCGA data, regardless of the origin of that data, be it like, the hearts, the brain, the pancreas, the endometrial, he found that you can classify all samples, all cancer samples, into four different tumor microenvironments. And these four tumor microenvironments could help you predict if someone could respond to immunotherapy or predict how long someone lived after being diagnosed with cancer. And this was interesting because it was cancer agnostic. So regardless of the cancer, basically you can study the cells around the cancer. So this is called the tumor microenvironment. So you're studying the cells surrounding the cancer, and you can break that down into four different types or classes of tumor microenvironments, regardless of what cancer you have. And these four different classes could tell you if a patient, for example, could survive longer, or if a patient, for example, could respond to a type of treatment. And the four different types were immune enriched, fibrotic. Immune enriched means that there are immune cells surrounding that tumor, and there are also some like fiber cells. There's immune enriched non-fibrotic, so there's just immune cells, no fiber cells, or no fibrotic cells. And then here we have, or fibroblasts rather. And here we have fibrotic only. So there is the immune cells. I mean, this is the cancer rather, and there are lots of fibroblasts surrounding the cancer. And then finally, there's desert. So there is the cancer tumor, and then there's barely any immune cells. And the reason why this is important is immune cells do attack cancer cells, and they're quite important in cancer treatment. In the case of the fibrotic, so like the presence of the fibroblasts, it appears that the fibroblasts form a protective layer around the cancer, making it difficult for like the immune cells to either like detect that the cancer is there or to even attack the cancer. So immunotherapy would not be very efficient. Then if you look down here, you can see a survival plot where like the dark purple right here corresponds to the immune enriched. We have the gold color right here, yellow gold. Then we have this color, and then we have this final color. And this is overall survival. So basically if you're up here, it means that the patient has good survival over the next few months. And he's showing here that the patients who have an immune enriched non-fibrotic have a better chance at survival or high chance of survival compared to the other patients. And then the patients with the worst type of survival was just purely fibrotic. So it's immune enriched non-fibrotic, and then a poor chance of survival will be fibrotic. This is important for deconvolution. Why? Because to classify these four tumor microenvironments, you have to go around the tumor and then look at the different cells that exists around that tumor, right? And in the case of bulk, it's an average. So if you can break down those cells into different proportions, then you can properly classify that cancer tumor into like this different four environments. So if for example you break down that bulk RNA sequencing and you realize there's a high proportion of fibrotic cells around it or fibroblasts, then you do know that that cancer belongs to this category, or that tumor microenvironment belongs to this category, and will be very difficult to treat using immunotherapy. So you can then try a different, more aggressive form of treatment, as opposed to like, wasting time trying to use immunotherapy for this. This is why deconvolution is important. Lots of researchers do it today. We have lots of bulk RNA, but this is one application of deconvolution. Again, here I have like why it's again we have lots of data available for bulk, and then single cell, like we talked about in the previous slide, single cell RNA sequencing is very expensive. This is my final example that I would give of deconvolution, but this is the work that we're doing over at the New York Genome Center, where we're studying ALS. ALS is like amyotrophic lateral sclerosis. It's a very bad disease, and usually within if you get diagnosed, usually within the next year or so, most people die from the disease. But with deconvolution, we can break down again the bulk RNA sequencing to study the differences between people with ALS and people without ALS. So we can see how, for example, percent like the lymphocytes differ between healthy people and people who have ALS. Then we can do the same for the different cell types that exist in the brain and spinal cord region. And this tells us like if a cell type is higher in general with people with ALS versus people without ALS. So this is a different application of deconvolution. Okay, so now let's go into the machine learning portion of this. I think that's what we are here for. So we've talked about, I think Sam earlier was talking about LLMs. There are a few classes of LLMs that are used specifically for yes,
Okay.
yes.
Hi. Okay. So can you give examples of not LLM-based models, but regular models, like point models that are used for the conclusion? For example, think about PTA. One of the examples of good examples for conclusion or non-negative metric factorization in the math. So, uh, what are the common? What is the common standard of RNA out in cloud foundation?
If I can repeat the question, I think the question was what the standard tools for this type
Deconvolution.
convolution.
Yeah.
I have that in the next slide, so maybe I should just start with that first. So I'm gonna do it's all the way here. Okay, so pay attention, just like forget about the right portion. Let's just look at the left portion. So for deconvolution, like I said, in case of the smoothie, right? So you have your smoothie, you do need to have the individual fruits, right? So because like if you have no idea what a strawberry tastes like, or what a blueberry tastes like, you can detect what is a strawberry in a smoothie, right? So in the case of deconvolution, you do need to have access to previous single cell scRNA, single cell RNA data. So you have single cell RNA data. The full profile of that is cell type, so the different cell types in that single cell data by gene, so the gene expression. So let's say a cell type can be T cells, and then you have like maybe 20,000 genes, and then you have like a general expression value of T cells across these 20,000 genes. A different cell type can be B cells or macrophages, and then you have expression information across the 20,000 genes for B cells. In most cases, researchers using like wet lab science kind of work would pick out markers that specifically define B cells. So let's say we know that for B cells, the gene markers are like, let's say, gene A or gene B, or for T cells, the T cell markers due to like what we like the work we've done in the lab, or like let's say these different genes, we can compile those T cell or B cell markers and create what we call a signature. So we have a signature that is now the different genes, right? The marker genes, and then the cell types. So B cells will have marker genes, and then T cells would have marker genes. And with the standard deconvolution pipeline, you then comparing computationally these marker genes that you've compiled with your bulk RNA sequencing. So you compare your bulk RNA sequencing with the standard genes, marker genes that you've compiled manually. Sometimes you can do it computationally, but most times it is manual. Um, so once you compare this to different types of computational tools you can use, so you by comparing these two, you can break down the proportions of the cell types in your bulk using these marker genes. It's a math problem, and it's been solved before. There are different ways of doing it. One way is non-negative least squares. So it's like just linear algebra. If you have like a mixture model or a signature model, and then you have of pure rather, and then you have a mixture, you can break down your mixture into components using that signature. There are different tools. The two of them that I've highlighted here are music and DWLS. If you have time, you can, go take a look at how these two work. But basically, you do need some single cell reference, and then you need your bulk. In most cases, sometimes, like in the case of CyberSort, you need an already curated marker, and then you're comparing your signature with your bulk and breaking down your bulk into cell type proportions. So this
I
is
have
a
a,
standard.
I have a follow-up question on this, Ali. So, what is the difference between the full profile and the signature? Because, I mean, by looking at this, it looks like you would still need some marker genes to be able to define, like, the full profile. I mean, the box under the scRNA on the left-hand side. So, like, what's the, difference between, the signature and the full profile?
So that's a good question. The full profile here is cell type, and then the full profile here, the gene is across all the different genes that are profiled in the single cell RNA data sets, which can be is I think with transcripts, sometimes it's 30 something thousand that we're working with. The markers here are just a list of genes for each cell type. So by the time you combine the markers with this, now you have your cell type, but as opposed to having the 20,000 genes, you only have a select set of markers that researchers have defined that defines that cell type. So it can be like 100 markers as opposed to 20,000 genes. So 100 markers are maybe enough to define what a B cell is. In the lab, sometimes like 5 markers, but this has to be compiled by someone. There are some tools that would do this compilation itself, but but either ways, you have to go from a full profile to a signature. So in the case of DWLS and music, it would create this for you. In the case of CyberSorts, the markers are already done, and then you can pass in those markers and it would create a signature for you. But the main difference here is size. So with the full profile, you're talking about the different cell types by like all the genes present in your single cell RNA-seq data. And then with the signature, it's just the different cell types again, but with just a few marker genes that have been compiled either computationally or manually. But one reason for this, and I was going to go back to this in the previous slide, is you can see that this is very computationally expensive or manually expensive. So in this case, you have to for sure know that what your markers are, and different researchers would like introduce bias to what a marker for a T cell is, and a marker for like an endothelial cell is. So that's one limitation of this. And then with this, it can also be computationally expensive. And With this too, one major downside is the presence of batch effects. So let's say we have a single cell RNA data that was sequenced here at the New York Genome Center, and we have one that was sequenced, at Mount Sinai, like a different research center. You realize that if you compare those two data sets, right, the same genes, the same type of tissue, there will be batch effects that separates these two data sets, even if it's the same genes, right, and the same tissue. So using this does bring a limitation since different single cell data sets have different batch effects, and those different batch effects can bring some sort of bias to your signature data set. I think I had one more question from Humphrey.
Yeah, I think Humphrey
Yeah,
asked the question. Yeah.
thank you so much, Ali. Please, I want to find out, you cited an example of maybe having data from two labs. Here, I want to find out if you have maybe single cell data from New York, where you are, and then someone has bulk RNA-seq data in Ghana, where I am now. Is it possible to be able to do something like this? Or can you leverage publicly available data sets, like from maybe two different labs entirely, one for single cell, the other for bulk RNA-seq, but then probably the same disease condition and the study? I don't know if this would, would be applied.
Yes, that's a great question, and you're right. Usually, you use different sources. So, like with all the work I've done so far, the bulk might come from the New York Genome Center, and then the single cell might be from a different lab. And we have some internal single cell data sets, but we also have lots of publicly available data sets, and that's the typical way you do it. So you get publicly available single cell to use for deconvolution for your bulk internal data. And the reason why it shouldn't really matter is the tools that are used in the standard deconvolution pipeline are supposed to like correct for batch differences between the single cell and the bulk. So for example, music actually requires that you have single cell data from more than one source. So let's say you have single cell data from Mount Sinai, you have single cell data from the New York Genome Center, and you have one from, let's say, China, a lab in China. You can combine that together and you can pass that information to the batch information. So the Chinese lab versus like the New York Genome Center versus like a lab in Ghana. And music will try to look for shared signature across these different labs that are still defining that particular cell type. So music would go in and say, does gene A does gene A define B cells? If so, is gene A um is gene A not variable, or is gene A, that's what I'm looking for, um consistent across the three different labs? So if gene A defines B cells, it's a potential marker. And then if gene A is consistent in this lab, and this lab, then music would like weight gene A very highly as like a potential marker for that B cell. DWLS does it differently, but most of the tools would like have ways to attempt to try to correct for those batch effects. So look for like markers that are just not markers for the cell type, but markers that are consistent across the different samples that you've passed. In this case, we pass different labs, but sometimes you can pass different patients because if I take single cell from myself and single cell from Sam, there might be batch effects just due to like different donors. So you can pass that into music, and it would attempt to like correct or assign weights, higher weights to genes that define cell type, and then genes are consistent across the different batches that you've passed.
This is great. I mean, if I may add, one more thing, because we've not like, really done single cell before. So on the right-hand side of Ali's, presentation, under the deconversation, right? So there is a grade where the rows are cells and the columns, are genes, right? So rows, cells, columns, genes. This is an example of a single cell RNA-seq, data sets, right? So you may have like, a bunch of cells, like depending on how you sequence it and where you sequence it, you could have about like 10,000 cells or even like a million cells with probably a fixed number of genes. And then the values Ali, if you could help us here, the values in each box would be um, the transcripts level, right, for a particular gene
in
for a
that cell.
particular cell, right,
Yes.
right.
We can come back to this, but I think that's a good example. So like I should have shown this the first slide. This is what a single cell data looks like. So the rows here are cells, like Sam pointed out. So cell one, cell two, cell three, and then the columns here are genes. So you have like and like all the genes that we've profiled, and then the value would be the count of that gene or the expression of that gene within that cell. So if that count is very low, it means that that gene is not really expressed in that cell. If that count is very high, it means that gene is very much expressed in that cell. And this would be a very large data set. So sometimes you have like 20,000, cells, and then how many genes you've profiled. But then what's important is if you have like these cell level information, you can either like manually or computationally annotate those cells. So we can say, oh, cell one is indeed a T cell based on the expression of the genes that we have. Cell two might be like a different type of cell, like an endothelial cell. Cell three might be something else, right? In the case of bulk, instead of like cell by gene, we have the bulk profile. So this would be like a sample, can be like sample from me, sample from Sam, sample from Humphrey, and then this would be like the gene, and then the value here will be also an expression. So but the difference here is since this is bulk and this is like, that average and combined across cell types, the expression here would be the expression of that gene across the different cells within that bulk. So all combined. And then bulks within the person here, within Ali's sample. And then bulk two would be, the expression of gene one across all the different cells within Sam's. And then the third will be for Humphrey. So they're two different types of, they're related, but they're two different. This is more granular and this is more like averaged out. But
This
we
is
have
really great. Yeah, this is really great, Ali. And like if I may interject you once again, I'm really sorry for doing this. I just want to make sure that like they are able to relate with it. If I may add one additional thing, right? So rows of cells, columns, are genes. It could be represented, differently depending on the tool you use, like R or Python. But then, I think at the end of the day, one thing that we are doing, or one thing that almost everyone, does, think of it, this way. The rows are cells, but then you want to tell, maybe if you think, like, cell, like the row one is for, lymphocytes, there are different, types of, lymphocytes, right? There could be like, CD8, positive, lymphocytes, and there are some genes that are, really, expressed on lymphocyte, for example, the CD8s. So if it's CD8, you expect to see like, CD8, type of gene, CD8A, CD8, B. But then like it could be resting, it could be very active, it could be, the CD8s could be exhausted. And you want to also use this, marker genes, to be able to tell like the functionality of the cell at, at the points of, sequencing. So is it, naive or resting? There are some marker genes. Is it, cytotoxic or actively, fighting, the tumor? You look for, marker genes, like, granzyme A, granzyme B, perforins, and so on and so forth. Is it exhausted? Is the T cell exhausted? You may want to look for, markers like, PD-1, CT, LA, 4, LAC3, so on and so forth. So, in terms of engineering, this is it. And, I think the marker genes, which, I think it's usually created by the community will help you, get a lot out of your data set. So Sorry for interjecting again, Ali. But I just wanted to, make sure we are all able to relate with the data sets we are seeing.
Laat ons nog een Nee, de beelden ooit, hè. Heb ik op zijn gooit weet net op. Beelden die man.
Okay. Okay. I'll resume then. Thanks for the explanation, Sam. So going back to large language models, so we've worked with lots of these already, but there are some classes that are specifically for this kind of work. So for RNA expression data, we call them single cell foundation models. I'm sure there's also like bulk foundation models as well. But basically, there's been an explosion of these models since we have lots of single cell data available. And how they work is we have databases that have different single cell data sets. Those are converted to, matrices. I'll go through this quickly because we'll come back to this again. They're tokenized, so to talk about tokenization in the next few slides. The different model architecture, so usually there's some sort of like transformer and encoder decoder based. There's pre-training, and then you can then use that pre-trained model for any downstream analysis. But this I rushed through because I want to like talk about this again. But here's like I have this just to show that it's again most of them are based off of like this single cell data sets. So the one we're using today, and I feel kind of bad I chose this one, because it's only available using the GPU, but there are others that are not available using the GPU. So like self-to-sentence does work with a CPU. It's quite heavy, but it does work. But GeneFormer has been published already, it's out of the archive, so I think it's a good example of what we could use. So the original GeneFormer was trained on 30 million single cells. So 30 million single cells. This is captured by that plot right, that picture you can see right here. Those different clusters correspond to the different cell types. So cells of the same cell types will cluster together. So 30 million of those, so that's a lot, right? So 30 million of the single cells, and you have this model that has seen 30 million single cells across different data sets, across healthy disease states, across different diseases. And the idea is, since this model has seen lots of different cells and cell types and different batches and variations, the model should be robust to batch effects, and then can be used for lots of different downstream tasks. So this I'll talk about later, but let's just jump right into the C portion of this. How this works is very similar to how ChatGPT or Gemini or Claude works. So for a given single cell transcriptome, so a given single cell, so this is one cell, we rank the genes based on expression. So we rank the genes in such a way that the gene with the highest rank is the gene that's like number one, has a high expression, and number two is unique, is unique to that single cell. So highly expressed, but not expressed everywhere. So you're ranking them based on normalized gene expression, and you're normalizing across the entire transcriptome that you have. So you rank them, and the reason why we do it this way is because the first rank should be the most important gene for that cell, and then the last rank should be the least important gene for that cell. So when you rank those genes, this is the tokenization portion of the work. You now leave the expression values behind. So before, we talked about gene A has an expression value of like a certain value, gene B has the same, but now all we have is rank. So gene T here is rank 1, gene H is rank 2, gene Y is rank 3, all the way to the bottom, right? And then the first rank should be the most important rank. Just like in sentences, if we have the genes ranked, we can like arrange them in a simple sentence. And then we do what we call max learning, where you basically hide some of those ranks away from the model, and then you're training the model to predict the ranking value of those genes that you've hidden. So like in a sentence, if it says in English, Ali is going to school, you can blank out, school and train the model to predict possible entries for that blank that you have. So it can be Ali goes to school, Ali is going to church, Ali is going to the mosque, Ali is going to the university. In the case of the single cell, it's very similar in that sense. But instead of like school, like we're blanking out the gene and the position of the gene. And the model is then tasked with predicting the position of that gene that you've maxed. And the idea behind this is if your model can correctly most of the time, predicts the maxed value of that gene, then the model can learn very complex gene-gene relationships. So the model can like learn that in this rank sentence that we've created, gene A has to be in the position of 6, and gene D has to be in the position of 7 for this single cell. And you do this across all the single cells that you have. In their case, they had 30 million single cells, but now I think they've increased this to about 104 million single cells across different batches, across different data, across different disease states, across different diseases. And once you have a robust model that can correctly predict these positions, these max positions, you can then extract the embeddings. The embeddings are just a vector that capture like this complex relationship. You can also extract the attention weights. The attention weights are also vectors, right? That the model is paying attention to while completing this task. And then finally, you can do predictions. So you can task the model to like predict if a cell is a certain cell type, if a gene is a certain, belongs to a certain class, different downstream analysis. It's slightly complex, but the goal of this slide is to just show that for each single cell, you're ranking the genes based on expression value, normalized expression value, and then you're tasking your model to predict the max rank of that expression value that you've hidden from it. And you do it like over like the 30 million cells, right? And the end goal is to like train the model to capture complex gene interactions or gene relationships across the 30 million cells. In this case, 30 million, but now it's 104 million. Here, the authors have like a breakdown of the sources of the data that they used. So a huge portion of it comes from the brain. They have immune, there's liver, heart, all the way down. So it's quite robust in terms of like origin of the cells. And then it according to the authors and according to the way the model is trained, it should be robust to batch effects. So batch effects like a sample was sequenced, let's say a brain sample was sequenced at the New York Genome Center versus a brain sample sequenced at KNUST. It should capture like brain signature regardless of the origin of that sequence. And it should remove genes, right, that are more or it should derank genes that are capturing batch effects, like the source of the data, and just focus on genes that are capturing complex brain chemistry, or biology rather, across the different brain data that you fed it.
So, I have a question, Lee. So it looks like you are getting, two things, for free. Because I mean, I haven't done this, before, but I heard that you have to do like, batch correction, each time you're doing, single cell, sequencing. But it looks like once you're able to, use this, transformer, model, it sort of like, corrects, for the batch, effects, um,
Yes.
in addition to, being able to glean, biological information. More like two for one,
Yes, that
but it's
that is correct.
computationally expensive, I can see.
It is computationally expensive, and they're getting bigger and bigger. So, but at least you don't have to like train them yourself. They've been pre-trained, so you can use the pre-trained version. But you can also like fine-tune this as well. I'm going to see. I don't know how much time we have, but let me see if I can get through some of the other stuff, so I can maybe show some code that I can share later on after the presentation. But here, outside of Geneformer, there are other tools that that do this. There's SCGPT. If you have the time, you can go and see how this works. You have cell to sentence, and then you have Cell Hermes. Cell Hermes is still it's still not fully published yet, but it's available on bioRxiv. And I'm still not sure exactly how this works in terms of like the training data that's used and the size of the data. But with these three, Geneformer now is 104 million single cells. The model size is about, 40 million, 10 to 40 million parameters. Cells to sentence initially was 410, but Google worked with cells to sentence to scale this up to 27 billion. So this one is quite huge now. But you can work at the CPU level with Cell Hermes and Cell to Sentence. So while this talk is mostly based off of Geneformer, that's GPU restricted, you can do the rest without GPUs. They're quite huge to download and run, but you can definitely play with this once if you're interested. Here I try to capture like again what exactly is happening. So the Geneformer, zero-shot model, you rank the genes, you hide the rank, and then you task the model to predict the rank that you've hidden. So in this case, gene 1, 2, 3, it should predict, gene 4 is the gene 4 is there. And with a fine tune, this is the zero shots general model that's available right now on Hugging Face. And that's how they trained it. There are no annotations. They just trained it to predict maxed or like hidden genes during that pre-training process. In the case of the fine-tuned version, it's your call. You can fine-tune on different things. What I've done so far with my work is I've fine-tuned to predict cell types. So let's say we have a single cell data set with annotated cell types. So endothelial cells, epithelial cells. I then further task the model to be able to predict this correctly. And I'll show some results for this. And then you can extract the embeddings one. So embeddings is one. Then you can also extract the attention weights. The attention weights should tell you what genes the model is paying attention to while it was being fine-tuned, and I'll show some examples in the next slide. And this is an example of fine-tuning. So what we have here is a scatter plot, 3 different scatter plots. On the x-axis, we have the mean attention. So I go into the model and I get the attention weights. I think I have code for this as well. So the attention weights on the general model that's available on Hugging Face. And then when I fine-tune that model as well, I also get the attention weights for the fine-tuned model. And basically, if you plot the attention weights that are the mean attention weights on the zero shots, so the general versus the fine-tuned model, you can tease out what genes the model is paying attention to paying more attention to in the fine-tuned model. So the straight line would be like this equal attention within both. But then if it's up here, it means it's higher in the fine-tuned and then lower on the zero shot. So somehow in the task of like predicting cell types, these genes are important in predicting cell types. Same for this, same for this. And I have this because like the original data, these cells were annotated as smooth muscle. These ones were annotated as myeloid. And then finally, this was epithelial ciliated. So basically what I'm trying to do here now is to show that you can basically go in and take those top genes that the model is paying more attention to in the fine-tuned version, right? And so here we have like FHL5, DST, CLD1, and you can basically pass this into like Gemini or ChatGPT and say, just given this 5 genes, right? What single cell type do these gene markers point to? That's all I'm saying. Just this, right? And I have this, colored. So this is the Gemini says it's smooth muscle, which is what the researchers already said for this as well. This case, it didn't get it correctly. Gemini says it's like T cells using just these genes. Researchers said it was myeloid. And then here, but I think myeloid are a type of, maybe not. But here it says ciliated epithelial cells, and researchers said it was also epithelial ciliated. So what this is showing that you can just take in just the top genes the model is paying attention to, and you can get the same annotations that people have, looks at biology to get to. The point here is that there might be the model is actually doing what it claims to be doing. It can learn very complex biology, and you can basically just use a very small version of that data that's been output to like predict the cell type. This is not the point of this presentation, but this is an application and to show that like the embeddings and the weights and embeddings are not completely random. There is some relationship between the embeddings and actual biology. Okay, so now going back to what we came here for, this is deconvolution again. I think it's been like circles, but we're back in deconvolution. We've talked about the standard deconvolution, and then finally deconversation, which is our tool here that we're using to like attempt to replace or compete with the different standard tools that we have here. So with this standard, we talked about the issues of like batch effects. With like deconversation, we're leveraging single cell foundation models. So here we can use GeneFormer, we can use cell to sentence, we can use SCGPT. And what we're doing is we're passing the single cell data into the foundation model. We're passing the bulk into the foundation model, right? We're not selecting any markers. We're just passing into like the general or the fine-tuned foundation model that we have. We're extracting embeddings. So at this point now, the single cell becomes cell by embedding space. The bulk becomes bulk by embedding space. And they're now in the same space. So before you had lots of genes. For GeneFormer now, you have 1,000 embeddings. So we go from 20,000 or 30,000 genes to just 1,000 embeddings for the single cell. And we do the same for the bulk. So 20,000 genes to just 1,000 embeddings for the bulk. And then we pick the same or a similar deconvolution solver. So it can be non-negative least squares. And then attempt to do the same thing that music does, but instead we're now working with the embedding space. So at the very end with the standard tools, you have sample by cell type. So you deconvolve this into sample by cell type. And then here as well, with deconversation, you deconvolve this into sample by cell type. The main difference here is we're using the foundation models, one. Two, we're not picking genes, right? We're just using the embeddings from the foundation models. We're trusting the foundation models to like, one, remove batch effects. Two, to like bring them on the same plane so we can compare them. So there's no need for like selecting batch effects or correcting for batch effects or selecting markers. You can just pass in your single cell data and your bulk data into your foundation model, and then you can use different linear algebra tools to deconvolve. But the goal here is we're bypassing this complex process right here. Okay. So proof of concept. So how do we know this works? We have data sets that is like, we have bulk data sets where there is accompanying ground truth. What that means is for each bulk data sets, we do know how many cells or the proportion of cells that went to create those bulk data sets. These are all public, by the way, so you can access this if you want. So we can then do deconvolution using the standard tools, and then do deconvolution using the foundation models, and then compare how these two compare against each other in terms of like accuracy at predicting the proportions of cell types. The next slides would show you what I'm talking about. Here's an example of a PBMC data. So it's a real bulk data. And GF here stands for GeneFormer. Cell to sentence stands for is a different model, foundation model. Music, DWLS, and Bayes Prism are standard deconvolution tools that we use in research. So Music, DWLS, and Bayes Prism are from here, while the other one is from the foundation models are from here. So what we're showing here is we're comparing the correlation of the ground truths to the predicted, and then we're comparing calculating the root mean squared error. So in the case of the correlation, you want to have a high correlation showing like that model is working, and then you want to have a very low error. So in the case of the foundation models, we do see 0.74 for gene-former correlation, 0.93 for cell-to-sentence, and then with the standard, we see 0.33, 0.30, 0.46. So basically here, the foundation models are outperforming the standard deconvolution tools. To point out here, and here's a summary of the different data sets we've tried this on. So the x-axis, we have the different tools we're using. Music, DWLS are both standard tools. And then we have the gene-former, cell-to-sentence, cell Hermes. And what we're showing here is across the different data sets that we have, the foundation models seem to be doing quite well across the different data sets. In the case of music, so music does well sometimes it does quite poorly, and DWLS is also quite good, so it does quite well on 5 out of the 6 data sets. But with cell-to-sentence, you do see just good performance across all the 6 data sets that we have. If you combine all the results together, you can see that here we have mean correlation, so you want to have a high correlation, and then you want to have a low root mean square error. So here we see that cell-to-sentence, the fine-tuned version does quite well. DWLS also does quite well. This is like a standard tool, but then we do see gene-former here as well. So we do see that there is some high performance associated with the foundation models, that the like music and Bayes Prism, also foundation models, standard models. I mean, standard models are struggling while the foundation models are doing quite well. Only DWLS comes close. So only 1 out of the 3 standard tools we tested came close. But the rest are struggling across the different tasks. So this I'm going to skip, and then finally we have this where like it's computationally expensive for inference, regardless, and for fine tuning. So here we're showing the number of samples passed, and then the time it takes to extract embeddings. What we're showing here is that with the red curve right here, cell-to-sentence CPU is possible, but takes a very long time to extract embeddings. So for one sample, it's like 68 seconds per sample. Cell Hermes also works on the CPU as well. It takes about 150 seconds to extract embeddings from one sample. And then the rest are all GPUs. With the GPUs, it goes quite fast. The blue is Cell Hermes, quite fast, 8 seconds per sample. The pink here is Geneformer on the GPUs, quite fast, 8 seconds per sample. So they're quite fast in general, but once you get to the CPU usage, it does take time to extract embeddings from the CPU, but at least it does work. It is possible to like download these models, so cell-to-sentence and cell Hermes, and start playing with them in terms of extracting embeddings from them. That was my very last slide. I do have some code I would like to run through. So here is questions while I prep for, we can have a discussion while I prep for what I wanted to show. So please interrupt again if you have questions about like the models itself, how to download them, yeah. I know Humphrey had asked one time about data sets, so single cell data sets. I was going to point out
Yeah.
cells. I'm not sure if you've used this tool already, It's a free resource by Cell by Gene. I think is funded by Chan Zuckerberg Initiative, where they've compiled lots of single cell data sets available publicly. So if you're going to try out these tools with a different type of data, single cell data, you can come in here and download any of them. So you can explore them. For example, let's say I cared about this one. If you click on explore, it would show you how you can also download it as well. But then if you want to explore, it would show you like the different cell types that exist. It's taking time now, but it does work. So you can cluster the cells, the different cell types that exist. Usually you can color by, maybe it's my Wi-Fi, you can color by different variables. Like cell type, for example, you can color by cell type. But yeah, you can get any large single cell data sets. So I was going to point that out before I finish today.
Yeah. Thank you so much. Thanks.
So, Humphrey, if I could also get this, So for each single cell foundation model, you are getting or you are extracting the embeddings for that, foundation model. For example, the genoformal. You are taking the entire embeddings of that foundation models.
It will be your hands-on class on Monday. Okay, so you will.
Sorry. So I was asking that, for each single cell foundation model for this application, you take, maybe a particular single cell foundation model, and then you take the embedding that comes with it. Or you are embedding your cell types to the foundation models, for example, gene farmer.
Yes.
Do you get my confusion?
Wait, can you repeat that again?
I was asking whether you get my confusion.
No, I don't. So can you can you please
So I was trying to see, or maybe it'll come up, in the code. So each of the foundation models, have and have embeddings associated with them, I guess, right?
mhm.
So for this application of what you're doing, you take the foundation model and then you take the corresponding embedding that comes with it. And then you do something with it, which the code is going to show us for each foundation model. Is that the case, or something
Yes,
else?
yes,
Okay.
yes. I will mostly focus on uh I'll mostly focus on just working with GeneFormer itself. But yes, that is right. For each of them, you can get an embedding and use the embedding however you want to use, however you want to use that embedding. That makes sense. But how about I jump to some code so we can, my screen is paused, so hmm, okay. Okay, so some of this I've ran already because it takes some time to run, but just to give you an idea how this works, and I can like have a cleaner version of this available for download soon. But basically, GeneFormer is on Hugging Face, so this is what it looks like. You can go to model card, you can go to file versions, and under file versions, you have on the file versions, you have different examples of how it's being used. So in the examples, it does show you how the model can be used for cell classification, for extraction and plots in cell embeddings, for gene classification, for in silico perturbation, and all of that. So this is public, and you can get this, and then there are community notes. or questions where people have issues. So if you have issues, you can post yours on here. For my tool, it's called Deconversation. So it's a PyPy package, and it's also public as well. So if you want to play around with this, but I do have a snippet of code and just like working specifically with Geneformer. It's a Python notebook, and so with Geneformer, like let's just go through, and most of these models are the same. Go through like the pipeline and how these work. So we have, I'm importing my different libraries that I need. Here, the first step of Geneformer is to tokenize that data. So you import the tokenizer. And with the tokenizer, you'll find in the tutorial as well, you get to define the important variables in your single cell data. In my case, we have, final type, final lineage, cell index, cell ID. And the tokenization function is just a single function. So you pass in the path to your single cell data. This should be, I have this right here. This should be like a H5AD data set. So for single cell data sets, you pass in a path to like your output path. So where you want the data to be stored, output directory, and then it's just a prefix. So I just saved mine as tokenized data. And then the file format is H5AD since our single cell data is in H5AD. And again, you can get H5AD data sets publicly from CZI cell by gene that I just showed previously. So once you've tokenized the data, this is doing the, ranking. I have that here, ranking the gene. So at this point now, you no longer have expression. You just have the ranking per each cell, right? The next thing you want to do is extract the embeddings. So you can extract embeddings directly from the zero-shot model, which is what we're doing right here. So you load your embedding extractor, and then you define the labels that you're interested in. So final type, cell ID, final lineage, cell index. And then this is the embedding extractor right here. So I'll label this embedding extractor. So Steve Theodoros is the author of Geneformer. So this is a path to the Hugging Face model. So this is the general model here. So your model goes in here, can be your fine-tuned, but in this case, the general model. And then we have the path to tokenized data. So this is what you tokenized here. You pass that in, you pass the path to your output, so like where you want to save your data, and then a prefix to just put as a prefix in front of your output. So here we've gone through like tokenization. This portion is embedding extraction. And then here, we're loading the single cell data. So that's what I have here. So you can get yours from cell by gene. And if you run this to this point, you have your embeddings extracted. So embeddings should be extracted at this point. So here I'm loading my single cell data, and then here I'm loading the extracted embeddings I just extracted, right? They look like this. So here we have cell ID, so these are individual cells, and then 0 all the way to 1151 are the Geneformer embeddings. So now as opposed to like cell by gene, you have cell by embedding space. So 0 all the way to 1,151 embeddings. You can cluster this and play around with this as well. But in my case, I'm going to add this embedding to my single cell data just so I can have a single data set. So I add the Geneformer embeddings to my single cell observation data, and then I cluster that. So this is what embeddings look like. Again, this is purely embeddings, not expression. I cluster this and I color by the different annotations that we have. So the embeddings, again, in this case, do create clusters that are definitive of like those different cell types that we have. So DSCs, endothelial cells. Yeah, so it does work. This requires a GPU, but the process should be the same with SCGPT, where like you download the model, you tokenize the data, you extract the embeddings, and then you can do whatever downstream task you want to do for the embeddings. You can use embeddings to build like a random forest model, if you wanted to do that. But I think these steps are just, general and quite important for any downstream task. I also have some code for fine-tuning, specifically Informer. So again, you load your single cell data. I'll properly label this before I share it out. And then you have here's this filter dictionary that Informer allows, where you can like subset to a few cell types that you're interested in. This is just mine. Yours can be, shorter than that. Let's say in your single cell, you only care about these 4 data sets. That's fine. You just do that only, as opposed to working with all of the data. Here you define your training arguments, and these are like more standard across like different LLM-based technology and transformer-based technology. And this you can Google to see what each of these stands for. And finally, here you define your classifier. So in our case, we're fine-tuning to predict cell types. So with fine-tuning the models, we can properly predict these different cell types, which is a very easy task for the model to do. So here is you set up your classifier. Final type is a cell type here in my case. And then here I also have code for like subsets in the data. In machine learning in general, sometimes if you have like an over representation of one class versus the other, so let's say you have 1,000 T cells and just 200 B cells, that can affect the performance of your model. So I have code here showing you how you can subset to just a few cell types. Or you can balance rather the cell types. So as opposed to having 1,000, you have like 500 and 200 B cells. That way you don't have a lot of class imbalance. So here I have code, you don't have to run this, but here I have code showing that how you can like try to like stratify and balance out your different cell types. The model does fail if you have very severe class imbalance. And then final few lines of code I will share would be you prep the model, right? And then you run, this is a fine-tuning portion of the model. You fine-tune the model using the preps data sets. So, and then here's Informer code for evaluating the fine-tuned model for your different task. So all this I'll share, and I'll add like really guide on like what's going on here, so it can be replicated. But yeah, with all of this, you can have a full functioning, in this case, Informer model. And yeah, but again, you have cell by gene for different cell types. I can try another one. Let's see, PBMCs, lymphocytes, but just to show you that there is definitely more. So myeloid cells from human PBM cells collected in Indonesia. Let's try that. That's a lot of cells. So 70,000 cells. Explore. While that's loading, I'm going to close this one here. Again, I have my decomposition, so you can check this out if you're interested. And then here, finally, we have how like Informer does work. So again, you can read the manuscripts to better understand all I explained about the tokenization, the ranking, max learning. Here I have files and versions. So the different versions here, I mean, not I, they have files and versions, so different versions of the tools of the models that they have. So the original model was 30 million. That's this one. But now they have a 10 million model, a 104 million model. The 104 million model was fine-tuned just for cancer. So different fine-tuning models. And then they have a community here where if you look at closed discussions, lots of different problems. So GPU requirements that people had, embedding bulk RNA-seq data, which is what I try to do. So just different issues. So let's go back to this. I think it's loaded by now. So this is not a very good one, but you can color by cell type. You can color by developmental stage. You can color by donor ID. There's definitely clustering by donor ID. So that's a major batch effects in this data. You can color by sex. There might be some clustering by sex, I'm not sure. Then just different metadata, and this you can definitely download and then use for any downstream tasks across the different models I've mentioned today. That's all I have. I promised Humphrey that I was going to clean this up and then share this code so people can try this out. I'll also include like links and paths to codes for SCGPT and Cell Hermes as well. Those ones are available on the CPU. It's gonna take a lot of memory, but you can definitely do it. Then I saw a post, someone created like an API where, or like a light version of GeneFormer where you can, or I think it was GeneFormer, maybe Self-to-Sentence, where you can basically extract embeddings quite easily. So I'll share that as well. I think this should be enough guide with these and then this. Um, if you want to play with yours.
All right. Thank you very much, Ali. Yeah, it was really great having you. I don't know if we have some questions from people. Yes, but I really appreciate even the data set repositories you showed. I think trying to get data is quite challenging. Yeah, but I want to find out some few things. First of all, does sample size really matter when we are trying to do this? Can you just take two studies? Okay, I've seen this study. They've done one publication, brought this study, let's say, for 100 samples, 100 participants, bulk RNA. And then you see another data set somewhere, and you're like, okay, this one has like some 60 single cell. Is it possible to put these two together to do some of these things?
Uh, can can you repeat that again? So put what's together again?
Like I'm trying to say to replicate an analysis like this. Is it
Yeah.
possible to, like, should we consider sample size in terms of maybe participants whose samples were sent for bulk RNA sequencing, and then maybe have others for single cell, or you need multiple bulk RNA-seq data sets and multiple single cell data sets. But I'm
Oh,
thinking, can
yeah.
one each do, or you need like
For
a whole batch of
yes, yeah.
data sets?
That's a good question. For the standard tools, you do need at least single cell from different donors, so you can just be one donor. But for bulk, it can just be, bulk is done at sample level, so it can just be one sample. It doesn't have to be more than one. And there's lots of, even if you don't have samples, there's lots of publicly available TCGA samples. So I could have shown you how to get TCGA, but I'll include that in the like file I'll send to you, where like you can just go in. There's a couple sites that host TCGA samples. I'm not sure if it's like geo blocked, but I think it's public for everyone. And you can go in there and, like, let's say you care about breast cancer, it's like a TCGA breast cancer cohort. I think it's about more than 300 samples. You can download that. If you care about pancreas cancer, that exists as well. So you have both access to, like, you have access to both single and bulk publicly. I think there's been some shift to like the democratization of these data sets. So you can definitely have access to this. But that was a good question.
All right. Yeah, Ali. So the another one from me, yeah. Would different experimental protocols, procedure actually not have an impact on the findings? Because I think maybe a way a lab is going to process the samples and all these things is actually going to talk about how the samples are going to be managed before even doing the libraries for sequencing. I am thinking different individuals, different expertise, different methods applied can also include some biases or some interference. And if you want to put these together, is it really going to rule out the biological? I'm trying to look at maybe the biological concerns from maybe the scientific community. They'll be like,
Yeah.
okay, different experiments different people, different protocols, and then we want to do this. Can that influence the
It can.
findings?
It's always an issue with these kinds of work. So there's always batch effects from different things, how samples are processed, if they're like fresh frozen or they're like fixed with like FFP, all that is always a problem. But you can't always completely correct for it. So that you can, if you write a paper, you put that in your your disclosure section that, hey, we tried our best to correct for this. There are different tools for correcting for batch stuff effects like that, like combat seek and the rest, but you can't always fully correct for it. But yes, you're right. That stuff, things like that will always affect your data. It's why you have to visualize on PCA first. So visualize on PCA first, do all the QC. If you think it's just too much of a hassle in like merging them, then maybe merging them is not the right step to do. But no, that's also a good question. I
All
do
right.
see someone asking if it works for human cells. Gene Former is just for human cells, but cell to sentence is human and mouse cells. Mouse cells. There is a different tool called transcript former that I recently heard about that was trained on lots of different species, I think 15 species, including C. elegans. So you can also try out like foundation models and not necessarily these ones have pointed out. But the ones I've pointed out are mostly human. But cell to sentence is human and mouse.
Thank you so much, son.
Yeah, I'm worried that I have a meeting clashing with this very soon, so I have to like hurry. But I will
Okay.
share lots of materials after this, and then have access to my email or my LinkedIn. If you have any questions, please feel free to reach out to me. But this was so much fun.
All right, thank you so much. And then we'll try and reach out to participants as well with a follow-up email. Thank you all for joining, and then we appreciate your time. Look forward to meeting next week, same time, 2 p.m. And then
Right.
enjoy a lovely weekend. All right, Sam, over to you.
All right. Thank you. Yes, so much again, Ali. I know you got to go. I just want to mention that you could try Google, Colab, as well. In the past, I think they had generous offers for, GPU and also CPU, as well. But if you want to like try your hands on, some of these, notebook, see if you could use it in Google, Colab, first. And I'll do more research with regards to, compute accessibility in the region, as well, maybe India and also, Africa and other countries as well. Yeah, so that's one thing I wanted to say. We'll send the scripts to you once we get there. So thank you so much. Officially,
Yes, thank you
we
so much.
we are done. Yeah.
Yeah. Have a
If you
lovely
want to
weekend.
stay on for the vibes, like we could do like 15 minutes, just vibes to discuss what's going on like in the AI world,
I thought we were
research in
taking
the world. Come again?
I thought we were taking a group picture.
Oh, okay, all right. So let's take the group picture, first, and then we could,
Yeah.
do the vibes. All right. So if you could really
Uh,
turn your camera on, briefly, I think this would be very
yeah, I can.
helpful.
So, when
Shadow, Christina, Bismarck. We want to take a group picture for branding, so we could get more credits and OpenAI credits.
all right.
Bismarck, Irenaeus, Nyami, a quick camera so you could take in the photo.
Christine,
All
are
right,
you
hi.
available to give a talk anytime soon?
Sorry, I just
Yeah,
like
I'm asking you, are you available to give a talk
um let me
to
think.
explain?
I need to pick a topic probably. I haven't thought about that, but it would be interesting. I work with pangenomes, structural bioinformatics, so I can think of something.
That'll be
All
amazing.
right, I'll reach out to you and see how we
Yeah,
can do
nice.
that.
Thank you.
Thank you. Cheers. Yeah. All right.
All right, quick one. Counting, up to 5. Countdown. We're going to space here now. 5, 4, 3, 2, 1. 0 G. All right, thank you.
Jeez.
Thank you.
All
Oh,
right. Thank you
someone
so
just
much,
came. Hi, Daniel. All right,
Daniel.
that's great. So we've officially closed. If you want to stay on for the vibes, and discuss, like, really what's going on, how you could use, these tools. We could do that maybe, in 15 minutes. And then, we could go do some work. So I think, one thing I would want to say, I think the build for Claude, science competition ended. Unfortunately, I didn't win. I didn't put in so much effort.
Oh,
That's fine. But I think the winner of the, build for science, something, I think,
okay.
he got an interesting, project. It's called Lazarus. And I
Wow.
think what he's trying to do is to resurrect, old, REPL. Because the problem is that, people, work in labs, they graduate, or they move on, and then the project dies, right? And, maybe 2 years, 3 years afterwards, you're trying to, install, get it running, and it never runs. Things change, right? So can you point Claude to like a GitHub repo and, you know, Claude will resurrect, this this repo and make it, live. Right? I think, that's interesting. I think he, totally really deserves it. And I was asking myself, hey, why couldn't I think of an interesting project like this? Lazarus.
Uh, People are
um,
thinking about great things, Oliver.
yeah, but
So
this is really applicable.
yeah.
Resurrects, old, yeah.
Some I was thinking we have we still have people on the line. Is it maybe very, I think it's going to be very nice if we could briefly introduce ourselves and see, because we are also trying to establish if we could find speakers among ourselves. We share ideas on what we are doing with AI as well. And
Right,
I tried out to Christina, right? And
right.
hopefully we are going to bring her on board, and then I think we can get to know about each other briefly, if you don't mind. Samuel, you can go for it, and then we take it up from there.
Oh, okay, all right. I wish you'd go for it, but leadership by example, so I'm going to go for it. So my name is Sam. I'm in New York City, not too far from, Lee. I can take a train and, see him. Still take some time, but not too far from Lee. We're in the New York Genome Center. So I'm in MSK, Memorial Sloan Kettering. We do cancer research and type of research. I'm also a student, right? And just like you and some people here, I'm really interested in how I can use these tools that are coming up with so much capabilities to one, finish my work, and also learn how to imagine, right? So I think for me, I think the greatest bottleneck since last year was that I think these tools are going to get better. They're going to get great and do amazing things, and you could just see that people are doing amazing things with it, right? And I think the bottleneck would be, how far you could imagine. So, these talks, are also, being, carried out to, broaden our idea in the space. I've seen Sven, spoke about, last week, and really learn how other things, are being done, get to know how, systems, operate, healthcare, systems, operate. I'm trying to see if we could get maybe like a director of a health institution to really tell us how healthcare works, from the top, to the bottom. And, people managing like very big, systems to really educate us on how things work. And then we could innovate, around. So, that's why, my blab so far. Some New York City student, and it's great to know you all and also learn from you. Yeah. Yeah. With
All
that,
right.
I pass
So
the baton to Humphrey. Yeah.
yeah. All right. So I needed to copy a lot from you and get the structure. All right. So my name is Humphrey Adi. I'm a PhD student with a Kwame Nkrumah University of Science and Technology, Ghana. I'm reading a PhD program in biodata analytics and computational genomics. Yeah, and then I must say that I have been doing some bacterial genomics, and quite interestingly, I've been able to transition to human genomics. And thanks to Irenaeus Nyami. So Irenaeus Nyami on the call right now was my student when I was a teaching assistant at UCC. He shared an opportunity with me in neurogenomics, and I was like, no, I don't like that. I don't like human genomics. And then gradually I find myself doing some stuff there. So that's really a good one. I am interested in learning how to build agents. I'm trying my hands on building some agents, building some bioinformatics tools, and then yes, in a couple of weeks or months, I'm going to be rolling out some interesting tools. And I'm still collaborating with people, friends, and experts on some of the projects, and I'm open for collaboration. Yeah, thank you. So since I spot Christina, I would want Christina to briefly introduce herself. But Drew will follow after that.
Yeah. So hi everyone, I'm Christina. I'm a research fellow in Australia, in Perth. So like I guess we are not located very close by, but very happy to join your meeting. I attended, I think, a previous meeting about Clow Bio and genomic AI with like previously a few months ago, and I actually enjoyed it. And yeah, I decided to join today. I work with plants and fungal pathogens, mostly to study disease, like how pathogens infect plants for crop and agriculture. And yeah, I think like I'm very cutting edge in a way that I work with data structures like pangenomes, graph pangenomes. Recently I started working for like structural bioinformatics, all the AlphaFold, ESM-fold, and also all the new like algorithms that came after that, so like protein interactions, and I'm very keen to explore that because I think there is a lot of potential. And yeah, I'm happy if anyone is interested to work in the same topics and like get in touch with me. Happy to get in touch with anyone and work with anyone. Yeah, and like looking forward to the next meetings. And like maybe I can think how to better use clothes for like in a little bit different way. I can still like give it a go. And yeah, very happy to see you here. So next one, I guess Bismarck. It's on my screen.
Okay, Bismarck. Bismarck, are you online? Hi, Bismarck. All right. Then I think I'll have to choose one person. Oliver
Hello.
Noah. All
Yes.
right, Bismarck is here. Olive. Okay, sorry. Bismarck, sorry. Olive. Bismarck.
Yeah,
Bismarck,
yeah.
your face. We want to know you.
Oh, okay. I'm Bismarck upon a health tutor at nursing school, and I'm currently doing my PhD program at KNUST in bio data analytics and computational genomics.
Great.
Great.
So, what are your research interests, if you want to say?
I want to move into genomic nursing, actually.
Okay. All right. Thanks. All right. I think he's muted again. Olive Noy.
So my name is Olive Noe, and I'm interested in metabolomics, specifically LCMS metabolomics. So I have a background in biochemistry and data science, and I'm trying to move into LCMS. I'm trying to move into LCMS, data science, metabolomics. Yeah.
Oh, okay. That's great. So are you currently doing some things with AI? I mean, projects with AI?
No, not at the moment. I'm trying to hop onto any project that's available.
Okay. All right. Thank you so much, and good to have you. How about Alan Waddell?
Hey, sorry I joined late. I
All
just
right.
saw it posted and I was like, oh man, it sounds cool. So jumped in. And I'm on West Coast time. So yeah, a little behind. But yeah, thanks for putting this on. So I am in New York City, currently traveling in Seattle though. Uh, and yeah, I've been in the game for a little while. I've been a computational biologist, bioinformatician for like past 15 years or so. I'm currently trying to figure out what my next move is. And yeah, given the advent of AI. Why are you giggling, man? Why are you giggling? Um,
Oh, great. That's great. Yeah. Great to
yeah.
hear from you. Yeah, and thanks for joining. Actually, even yeah, we appreciate it, your time. And thanks for coming.
Okay.
Yes. Please, is Daniel online? Daniel Safo. Oh, call ladies too.
Hello.
Yeah. Hi.
Um, okay, my name is Daniel Osapo. I
Okay.
currently work as a quality control microbiology analyst at
Okay.
Campus Pharmaceutical.
That's great. Thank you so much, and nice having you. Dennis, can you introduce yourself? Dennis.
Yeah, sorry.
Okay.
Hi, I'm Dennis Elom Kosina. I'm currently a teaching assistant with the Department of Biochemistry. I am very interested in bioinformatics. I'm actually learning. I've been learning for maybe the past 8 months with the help of the guys in the quarting lab, Humphrey, Shadow, and everyone else with Fosti. We are all learning, and I'm actually interested in the bridge between global health, some epidemiology studies, and I don't know. I'm trying to find a niche for myself right now. I'm not very certain yet. Yeah. But I'm very happy to learn, see how AI fits into the picture and all that.
Okay.
Thank you.
Arrhenius. Yeah. Thanks. Thanks for having me. Arrhenius, can you introduce yourself briefly?
Yeah, so thank you very much, Humphrey. I am Irinius Nyami. I am a recent graduate from my bachelor's, yeah. And well, I've been experiencing and experimenting a little with microbiomics. Yeah, but then I won't say I have much experience in there, but then there's some interest over there. But then I generally have an interest for neurogenetics, but I think I'm also developing interest for bioinformatics, and I would appreciate where to actually start from. Yeah, but thank you very much for this.
All right, that's great. Thank you so much. Thank you so much. Thanks for passing through. Faustina, please introduce yourself.
Hi everyone. My name is Faustina Frempor, and I'm currently an MPhil biochemistry student at KNUST. And I'm interested in forensic science and immunology and infectious diseases. And I currently picked up an interest in bioinformatics, so I've been learning. And with AI too, I'm trying to build an agent. So I'm interested in AI in fashion. So I want to build an agent that will help with virtual measurements so that even if you want to sew your dress, you don't have to walk all the way to your seamstress in the comfort of your home. You can take up your measurements, choose inspirations of designs, choose your fabric to use, and every other thing. So in the comfort of your home, you get to sew your dress, get it delivered to you. So yes, that's what I'm trying to do with AI. So yes, this is me. Thank you, everyone.
Yeah, thanks. So this is very great, I think. Some people say scientists are boring, and we have some cool stuff here aside biology, moving away from biology. Yeah, people can venture into so many things, so that's good. Good to hear from you. Drew, please, can you hear me? Would you want to introduce yourself? All right. Shadow, please introduce yourself and your interest. What are you doing in AI? Hello, Shadow. All right. I think Shadow is not around. Yeah, and can you introduce yourself, Kofi and Bade?
My name is Kofi and
All right.
I'm currently doing my master's in MPhil biotechnology, and currently I'm using AI to review most of my projects and a lot of computational analysis.
That's great. Thank you so much. And then I think we have been able to interact with everyone. Yeah, so Sam, over. I think this was a very great session. So we'll put together the scripts and everything that we are getting from Ali, and then we share them across. And we look forward to having you on Fridays, 2 p.m. GMT. I'll send calendar invites so you can actually check in on us anytime you're available around this time. Over, Sam.
And check out our website. I think our name
Oh,
looks like a crypto. I think the website looks like a crypto website, so I'm not able to open it on any of the campuses, MSK, Rockefeller, or something. But the website is agentic, ghx.org, .org.com. I mean, it all works. We own that domain. So yeah, I mean, check it out. I think I'm trying to see if I could, how do you call it, do like an AI generated, summary. Because I have these morning briefings, where I started recently where I tell Claude to, like scrape the internet for news about AI in biology. Where is the field, moving with regards to, so this is what I see. So there are third parties building on top of the frontier models, like in the cloud. For example, like Edison, by Future House, Philo or Binary. And then now there is also cloud science, right? So I think the frontier models have ambitions to build like in-house, pipelines for scientific discovery. So what I'm interested in is how is the gap really closing, and what are some algorithms development and so on and so forth. So I think maybe I would also share with everyone so that we could also like get the latest news. Yeah, but yeah, it's been wonderful to see you all and learn with you, and we hope to see you next week as well. Yeah.
Thank you very much.
The website. Yeah. Just.com. Yeah. So .org.com.
Yeah.
I think you may be able to access it. But yeah. All right.
Thank you.
So with that, you could say. Okay.
All right.
Have a great week.
Yes. Bye, everyone.
Bye. Bye.
Bye.
Bye.
All right, Humphrey, I think I'll talk with you maybe on phone.
Sure.
Bye.
Okay, bye.